Hello students, today I am going to start a new topic, that is chemistry of nucleic acids. Nucleic acids are important biomolecules with varied functions that are vital for life. They store, express and transfer genetic information. It is the expression of genetic information that determines whether one is a man or a mouse, or whether a cell is a bone cell or a brain cell. There are two classes of nucleic acids: deoxyribonucleic acid, that is DNA, and ribonucleic acid, that is RNA. DNA stores genetic information coded in the sequence of its monomeric units, while RNA is involved in the decoding of this information, resulting in protein biosynthesis. Nucleotides are the building blocks of nucleic acids. In other words, nucleic acids are the polymers and are composed of linear sequences of repeating units of nucleotides. We call it polynucleotides. The nucleotides, the nucleotide, the basic unit of nucleic acids, composed of three components. They are pentose sugar, it is ribose or deoxyribose, a phosphate group, and a nitrogenous base, either purine or pyrimidine. The combination of a pentose sugar with a nitrogen base results in nucleoside. Now, the sugars in nucleic acids, two types of pentose sugars are present in the, that is in the deoxyribose found in DNA and ribose found in RNA. The ribose, which has a hydroxyl group, has again this second position, sorry, second position, and C2 has only hydrogen in case of deoxyribose. Now, the phosphate groups in nucleic acid, the phosphate group of one nucleotide that is linked to the pentose sugar of the neighboring nucleotide through phosphodiester bridges that forms the central backbone with the bases oriented outwards from the side of the sugar phosphate chain. Now, the bases present in the nucleic acids, two types of nitrogenous bases, that is the purines and the pyrimidine, are present in the nucleic acid. The purine bases present in RNA and DNA are the same, the adenine and guanine. Adenine is, that is here 6 aminopurine, 6 aminopurine, and the guanine is 2 amino, that is 6 oxypurine. The numbering you see of this purine ring is, that is first ring, that is anti-clockwise, and then second again it is a clockwise ring. Okay. There are minor purine bases. These bases are found in small amount in nucleic acids and called minor bases, like these are hypoxanthine, call it xanthine, that is amphoxanthine, that is 6 oxypurine, 6 oxypurine, xanthine, that is 2,6 dioxypurine, hydric acid 2,6,8 trioxypurine. Okay. Now, the pyrimidine base. The pyrimidine bases present in nucleic acids are cytosine, thymine, and uracil. Cytosine is present in both DNA and RNA. Thymine is present in DNA, and in place of thymine, RNA, it is present uracil. A few other modified pyrimidine bases like dihydrouracil and 5-methylcytosine are also found rarely in some types of RNA. Now come to the nucleosides. Nucleosides are formed when the bases, nitrogenous bases, are attached to the pentose sugar. You see, these all the bases are attached to the corresponding pentose sugar by beta N glycosidic bonds. So the first carbon of the pentose sugar is attached to the nitrogen atom in the ninth position in case of purine, and nitrogen atom in the first position in case of the pyrimidine. Okay. So that is the beta N glycosidic linkage. And deoxynucleosides are denoted by adding the prefix small d, okay, and before the nucleoside. You can see, you see the nucleosides, the purine, adenine, the adenosine, and one guanosine. So purine bases the prefix, and that is suffix is sine, adenosine, guanosine, inosine, xanthosine, and pyrimidine bases the suffix is dine, uridine and cytidine. Okay. Next, the nucleotides, the nomenclature, you see, when a sugar in nucleotide ribose, it is known as ribonucleotide. If the sugar is deoxyribose, is called deoxyribonucleotide. And when there is a single phosphate in nucleotide, is called monophosphate. You see this adenosine monophosphate, there is guanosine monophosphate or guanylic acid, cytidine monophosphate, cytidinic acid, uridine, that is uridyl acid or uridine monophosphate, inosine, there is inosinic acid IMFP. And for this, for deoxyribonucleotides, the small d, it is used as a prefix for this. So DMP, DGMP, DCMP, DTMP. An addition of 2 or 3 phosphate turned into diphosphate like ADP, ATP, and like that, diphosphate and triphosphate. And also for deoxyribonucleoside phosphate, that is the small d is used as a prefix. Okay. And you can see that now the esterification occurs at the 5th or the 3rd hydroxyl group pentose sugar. Most of the nucleoside phosphates are involved in biological function are 5 prime phosphates. A prime is used to denote this carbon of these pentose sugars. To differentiate the prime numbers used to detect the carbon atoms of the pentose sugar, and to avoid the confusion with the carbon atoms of the purine and pyrimidines. And the 5 prime nucleotides are more oftenly seen. So when you use the AMP, it is taken as granted for 5 prime AMP, but when 3 prime variety is there, it is always written as 3 prime. Now the functions of the nucleotides. Nucleotides are got so many functions, like they are the building blocks of nucleic acids, the DNA, RNA. They are not just the monomers of nucleic acid, but also play a role in various biological function. ATP, that is adenosine triphosphate, is the universal energy currency of the living systems. ATP is not an energy storage molecule, it is the energy carrier and takes part in the energy transfer reaction. The photosynthesis in plants and the breakdown of metabolic fuels like carbohydrates, lipids, and proteins results in the formation of ATP from ADP. ADP levels regulate the rate of ATP synthesis in mitochondria, and that is called oxidative phosphorylation. Hydrolysis of the high energy phosphate bonds in ATP that yield about 7 kilocalorie per mole of the energy, and the released energy is utilized for various cellular functions. Now you see the cyclic nucleotides like 3 cyclic AMP and cyclic GMP that act as second messengers. Nucleotides also structural components of so many coenzymes of B complex vitamins such as FAD, NAD+, that is NADP, and coenzyme A. They act as carriers of certain metabolic intermediates of carbohydrates, that is for carbohydrate, that is like UDP glucose. For lipids, it is CDP acyl glycerol, and proteins like S-adenosylmethionine. UDP galactose, GDP glucose, GDP mannose, CMP neuraminic acid, GDP fucose, UDP xylose, UDP uridine diphosphate, galactose, N-acetyl galactosamine, and um, UDP N-acetyl glucosamine. These 8 molecules are known as nucleotide linked sugars. They are important constituents of glycoproteins. So, we have now we are just in the different bonds stabilizing the structure of the nucleotides and nucleic acids. We have seen these covalent beta and glycosidic linkages that occurs between the C1 of ribose or deoxyribose and the N9 of purine and N1 of pyrimidine, and forming the respective nucleosides. The 3 prime 5 prime, that is the phosphodiester bridges, and they are form this phosphate group of nucleotides linked to the sugar of the adjacent nucleotide through these phosphodiester bridges. And besides this, there is high energy acid anhydride bond that is found in this ADP and ATP, you can say, and this yield 7 kilocalorie per mole of energy for hydrolysis of each high energy bond. And we see the hydrogen bonds between, that is adenine and thymine, or between adenine and uracil. There is 2 hydrogen bonds, and between guanine and cytosine there is a 3 hydrogen bonds are there. Okay. And definitely the 3 hydrogen bonds between G and C that makes a stronger bond than the 2 hydrogen bonds between adenine and thymine and adenine and uracil. That nucleotides also, that you see, they absorb ultraviolet light. The conjugated double bonds of purine and pyrimidine derivatives absorb ultraviolet light, and these are pH dependent. At pH 7.0, all common nucleotides absorb light at wavelength close to 260 nanometer, that is UV range, and the mutagenic effect of ultraviolet light is due to its absorption by the nucleotides in DNA, and that results in the chemical modifications like mutations like that. There are some synthetic analogues of purines and pyrimidines. There are some methylated purines also. We have seen the some pharmacological properties like theophylline that is present in tea, caffeine in coffee, and theobromine in cocoa. Well, synthetic analogues of pyrimidines and purines and pyrimidines, by introducing certain structural alterations in the heterocyclic ring or the sugar moiety, synthetic analogues of purine and pyrimidine nucleotides and nucleosides can be produced that possess pharmacological properties. Now, mechanism of their therapeutic effects includes the inhibition of enzymes essential for synthesis of nucleic acids, or their incorporation into nucleic acids and disrupting their structure. You see this allopurinol, fine, and that is used in the treatment of hyperuricemia and gout. The 5-fluorouracil, this is 8 azaguanine, 6 markup-to-purine, the sulfidyl group, the markup-to group. So these are incorporated into DNA and just before cell division, and that blocks the cell proliferation, and that's why they are used as anti-cancer agents. Now come to this DNA, the nucleic acid, deoxyribonucleic acids. Now what is genome? We call it genome. That is a complete genetic information of an organism occurring in the DNA in each cell of its body. Every cell of a multicellular organism contains the same complement of genetic material allocated among different chromosomes, each of which contains a separate DNA molecule. Now, since many organisms like human, they are diploid, they contain 2 equivalent sets of chromosomes, one from each of the parent. Their content of unique DNA is half, that is haploid, of their total DNA. For instance, humans are diploid organisms having 46 chromosomes per cell, the haploid being 23. The 3 into 10 to the power 9 base pairs of human DNA are organized into the haploid of 23 chromosomes. And the exact sequence of these 3 billion nucleotides defines the uniqueness of each individual. Now come to the structure of the DNA. The double helical structure of DNA is first proposed by the James Watson and Francis Crick in 1953, and they got this Nobel Prize for its discovery in 1962. Now, this elucidation of the structure of DNA is considered to be a revolutionary landmark in the history of modern science. In fact, its advent marks the birth of new disciplines like molecular biology, genomics, and biotechnology. Now, the Watson Crick model of DNA structure. The salient features of DNA structure proposed by Watson and Crick are discussed below. This DNA is a right-handed double helix. The two chains of the deoxyribonucleotides coiled around each other in the form of double helix. Two helical DNA chains wind around each other on the same axis to form the right-handed double helix. The two chains are antiparallel. That means one strand runs in the 5' to 3' direction and the opposite chain is in the 3' to 5' direction. The terminal residues whose C5', that is the prime we have mentioned on this pentose sugar, the carbon of pentose sugar, and that is not linked to another nucleotide is called a 5' end, and the terminal residue whose C3' is not linked to another nucleotide is called the 3' end. The two strands are held together by the hydrogen bonds between a purine base of one strand and the pyrimidine base of the opposite strand. The ring structure of each base occurs in a flat plane perpendicular to the sugar phosphate backbone, resembling the steps on a spiral staircase. The base pairing maintains a constant distance between the sugar phosphate backbone of the two strands as they twist around each other. The hydrogen bonds are formed between a purine base and the pyrimidine bases only, not between the two purines or two pyrimidines. This specificity is imposed on the base pairing by the fixed location of the hydrogen bonds. If the two purines face each other, they would not fit into the space available, and two pyrimidines would be too far apart to form the hydrogen bonds. Each nucleotide base of one strand is paired in the same plane with the base of the opposite strand. The hydrogen bonds formed between the purine, that is guanine and adenine, and the pyrimidine, cytosine and thymine, that is GC and RCG. While the two hydrogen bonds can be formed between the purine adenine and the pyrimidine thymine, AT or TA, and guanine always pairs with cytosine, and this is known as the complementary base pairing. That is the specificity, the adenine with thymine and guanine with cytosine, and this specificity has tremendous significance in the DNA self-replication and transcription. So, this is the complementary base pairing. So, as the three double bonds are present between guanine and cytosine, so it is stronger than between A and T, who has got two double bonds. So, the deoxyribose and the phosphate groups are hydrophilic. The sugar phosphate backbone is hydrophilic in nature, hence they are on the outside of the double bond orienting towards the surrounding water molecules, and the hydrophobic bases are stacked inside. So, you see these two strands, that is one runs from the 5' end towards the 3', another strand from that is so 3' to the 5' end. So, they are wind around each other is a double helix pattern. You see this This is the backbone, the sugar phosphate backbone. You can see the sugar phosphate, and this is the base pairing nitrogenous bases. That is GC, that is the pyrimidine, uh, purine and pyrimidine between guanine and cytosine, and then adenine with thymine, the double bond. There are 2 hydrogen bonds and there are 3 hydrogen bonds. So this, coiling of two strands leads to the major groups, major, minor groups, and the major groups. So coiling of the two strands creates the major group and the minor groups on the surface of the duplex. And the proteins interact in these groups with DNA without disrupting the double helix. Now if you say the geometry of the DNA duplex, the width is 20 angstrom, and each turn of helix, 34 angstrom with 10 base pairs and of the nucleotide. So each pair has a distance 34 divided by 10, that is 3 point. With distance between each pair, that is 3.4 angstrom. So this is 3.4 angstrom, 3.4 angstrom. And because of their length, DNA chains are described in terms of base pairs. 1 kilo base pair is equal to 1000 base pairs. The two antiparallel DNA strands, they are not identical in either the base sequence or composition, but they are complementary to each other. That means adenine always pairs with thymine and guanine always pairs with cytosine, the opposite strand. So DNA duplex is stabilized by two forces. One is hydrogen bonding between complementary base pairs and non-specific base stacking interactions. The strand that is transcribed or copied into the RNA molecule is referred to as template strand or antisense strand or non-coding strand. The other strand is sense or coding strand. However, it is noteworthy that each DNA strand can act as a template for the synthesis of its complementary strand, and hence hereditary information is encoded in both the strands in the sequence of bases on either strand. Now come to the Chargaff's rules. The experimental data provided by Edwin Chargaff and his associates in the late 1940s contributed significantly to the understanding of the double helical structure of DNA. These data collected from DNAs of different species are popularly known as Chargaff's rules. And these rules as follows. Number one, the base composition of DNA varies in different species. DNA from different tissues of the same species have the same composition. The base composition of the DNA in species does not undergo any change with the organism's age, nutritional status, or changing environment. In all species, A is equal to T, G is equal to C, and A plus G is equal to T plus C, that is thymine cytosine. Now, if you see the different types of DNA, three major types of structural forms of DNA, and the Watson-Crick model that describes the bDNA he has described, you can see, and other types of DNA find the aDNA, and aDNA is the right-handed helix, double helix similar to the bDNA, and it has 11 base pairs per turn. bDNA has 10 base pairs per turn. But zDNA is left-handed double helix, okay, and it has 12 base pairs per turn, and the backbone runs in a zigzag fashion. That's why name is the Z. Now comes to the denaturation of DNA. What is denaturation of DNA? Now the disruption of the hydrogen bonds due to the change in pH or temperature causes separation of the DNA strand and loss of double helical form, and this phenomenon is known as denaturation. However, the phosphodiester bonds are not broken, and just reverse is the renaturation. Now, the DNA rich in GC, that is guanine cytosine content, has high melting point temperatures due to 3 hydrogen bonds than the DNA high in A and T because they have two hydrogen bonds. Denaturation. Now, renaturation is the process by which denatured DNA strands have their double helical forms restored. That is also called renaturation or reannealing. Now, come to the organization of the DNA. Similar to protein structure, like DNA is also there is some hierarchical structure, like primary structure, that means nucleotide sequence, okay, of this regular stable structure organized into secondary structure and the folding of chromosomes into chromatin groups like to or the higher organisms, and bacteria it is called nucleoids, and that is considered as tertiary structures. Now there is some supercoiling of DNA. DNA is coiled in the form of a double helix with its two strands coiling around the central axis. The coiling of the axis upon itself produces DNA supercoiling by superhelix. It is just identical to a telephone cable which is coiled. The coiled cable can further coil itself into a supercoil. Supercoiling is an intrinsic property of the tertiary structure of DNA in humans. In bacteria, bacteriophages and certain animal viruses, the 3' and 5' ends of DNA molecule are joined to create a closed circle with no terminal. Supercoils are formed when a closed circle is twisted around its own axis. The negative supercoils are formed when the molecule is twisted in the opposite direction of the turns of the right-handed double helix of bDNA. The energy required to create this is stored in the supercoil itself. Topoisomerases are the enzymes which help in the formation of supercoils. Now come to the nucleoproteins. DNA in human cells is organized in association with a number of proteins, mostly the histones, resulting in the formation of chromatins. Histones which are cationic bind with the strongly anionic DNA. Histone proteins undergo certain covalent modifications such as phosphorylation, methylation, acetylation, ADP ribosylations. These are some of these covalent modifications which play an important role in the structure and function of chromatin. There are different types of histone proteins, H1, H2A, H2B, H3, H4. They are usually organized into a histone octamer. DNA wound around a collection of histone molecules or octamer that form the nucleosomes. Nucleosomes serve the purpose of condensing the DNA molecule, making it a more compact structure. In fact, the DNA helix in the chromosome has a length that is thousands of times the diameter of the nucleus itself. Now comes to another term called junk DNA. The major chunk of DNA, that is greater than 90%, is transcriptional inactive, and that is known as junk DNA, which is usually associated with nucleosomes and it not digestible by the nuclease enzymes. One of the major challenges in the field of molecular biology is to discover the function of junk DNA, which is otherwise considered is non-functional. So, active DNA which is transcriptionally active, the DNA which can be expressed, that is genes, that is sensitive to the action of nucleases, so that is only about 10%. Some regions in DNA are hypersensitive and are known to contain control sites, and these active DNA play an important role in the regulation of gene expressions. Now come to this mitochondrial DNA. Now DNA, what we find, the DNA is found in the nucleus. Apart from the nucleus, eukaryotic cells also have other organelles such as mitochondria and chloroplast, which contain DNA. Mitochondrial DNA, which is also that in short mtDNA, is double stranded like nuclear DNA, but is circular in shape. mitochondrial DNA is inherited from the mother because the mitochondria are inherited from the cytoplasm of the oocyte. So, the mitochondrial DNA is inherited from the mother and the mitochondria from the sperm do not enter the fertilized ovum. So, since mitochondrial DNA is more susceptible, it is more susceptible to mutations. The accumulation of mitochondrial DNA mutations result in degenerative diseases like Leber's hereditary optic neuropathy. That is an example of such mutations in mitochondrial DNA characterized by sudden onset of blindness in young males due to neuroretinal degeneration, and the disease is transmitted maternally. Now, DNA in prokaryotes. Now, bacterial DNA is organized into compact structure known as nucleoids. The entire DNA in prokaryotes is organized as genes and regulatory elements. However, the eukaryotic DNA is organized into a much more complex structure. Now, come to some terms like genes. A gene is a portion of the chromosome that determines a character or the phenotype. It is that part of DNA that codes for protein or an RNA. Coding and non-coding regions or sequences in genes. There are several intervening segments of DNA which do not code for anything, and such non-transcribable areas of DNA in genes known as introns, and the coding segments are known as exons. Almost all genes are composed of both introns and exons. Genes coding for histones have no introns. A typical gene has much more introns sequences than exons. On the whole, just one only 1.5% of total DNA is a coding, that is exons. The exact function of introns are not known. Now come to these centromeres and telomeres. No, that is, there are a few short sequences less than 10 base pair long in the human genome which are repeated millions of times per cell. These simple sequences do not encode proteins or RNAs. They are associated with certain structural elements of chromosomes, and these are the centromeres or telomeres. Centromeres, you know, the nucleoproteins which link the chromosomes to the mitotic spindle during cell division are anchored to the specific region of the DNA, and these are known as centromeres. These are centromeres. Okay, and this ensures an equal distribution of the chromosome sets to daughter cells. The sequences forming the centromere are the rich in adenine thymine pairs, AT pairs, and the telomeres, which are guanine rich sequences at the ends of the eukaryotic chromosomes. And they are organized through, actually, telomere DNA is synthesized and maintained by an enzyme known as telomerase. So, telomeres DNA is synthesized and maintained by an enzyme telomerase. The somatic cells of multicellular organisms lack telomerase activity. However, the germ cells have active telomerase function. The loss of telomerase activity allows the gradual shortening of the chromosome with each cycle of DNA replication and cell division until they reach the senescence. That is a stage at which there is been no more cell divisions. And perhaps this is the basis of aging process, the loss of telomerase function, and on the contrary, increased telomerase activity results in uncontrolled replication, abnormal cell growth, causing cancer. Now, considering the occurrence of extremely short telomeres in a rare disease called progeria, in which feature of old age occurs during childhood, an interesting question may arise: is there any hidden advantage to multicellular life forms in losing the telomerase function in the somatic cells? Considering the fact that active telomerase causes uncontrolled cell growth resulting in cancer, and aging could be a protective phenomenon that safeguards the organism from uncontrolled growth, that is cancer. Now come to some enzymes called nucleases. Nucleases are enzymes that are capable of degrading the nucleic acids. Endonucleases, the nucleases which cleave The internal phosphodiester bonds are known as endonucleases, and those that cleave nucleotides at the terminal point, they are called the exonucleases. Endonucleases are capable of recognizing specific sequences in DNA. They are called restriction endonucleases. So, endonucleases that are capable of recognizing specific sequences in the DNA are referred to as restriction endonucleases, and these are widely used in the recombination DNA technology. So, finally, the chromosomes. Chromosome is the storage format of genetic information of cells in all forms of life. DNA is organized. Chromosomes are visible under light microscope when the cells are suitably stained. The number of chromosomes in an organism is species specific. Humans have 22 matching pairs of chromosomes plus X and Y sex chromosomes. DNA is organized into chromosomes, and the protein DNA interaction organize chromosomes into the compact structures.
Thank you, students. Today I am concluding the class up to this. In next class, I will start with RNA.